Hello, everyone. So this is the second video in the series for Chapter 5. Here we are going to look at the first part of Section 5.4, and we are covering the endomembrane system, and we will be looking at the individual compartments and the specific processes that happens in each of these compartments. Let's go through the learning objective of this section, section 5.4. The overall objective would be to describe the structure of the endomembrane system in eukaryotic cells and summarize the functions of the different parts. So we can break this learning objective down into smaller pieces, which is very important for you guys to remember because, as I've said, it is these learning objectives that would be our basis for formulating our questions for the test. So the first sub-objective in 5.4 is to explain the roles of the nuclear envelope in regulating which molecules move in and out of the nucleus. The second is to relate the different parts of the endoplasmic reticulum to their roles in the manufacture of proteins and lipids. The third is to describe the roles of the Golgi apparatus in introducing chemical modifications to proteins and directing them to their final cellular or extracellular location. And the fourth in this part A section is to describe the role of lysosomes in recycling cellular components. And there will be two more learning objectives under 5.4 in part 2. So let's jump in and have a closer look at how these learning objectives are addressed in this lecture. So first, let us establish a big picture view of the endomembrane system in eukaryotic cells. Now, many organelles, and when we say organelles, these are membrane-bound entities inside the eukaryotic cells. They are not distinct isolated entities, but instead they communicate with other organelles. And they can communicate in two ways. First, their membranes are either physically connected by membrane bridges, like the one illustrated here or depicted here in the figure. For example, if you look at the purple region here in the nuclear membrane, okay, you can see a physical connection between the nucleus and the next organelle, which is the endoplasmic reticulum. Second, the organelles can also communicate by budding off and fusing vesicles that transport substances between them. In this illustration, the budding off is illustrated here, and the fusing is illustrated in this. So you can see that the movement of molecules, for example, between endoplasmic reticulum and the Golgi apparatus, which is this red organelle here, is by the budding off and fusing of vesicles that transport substances between them. So this system of interconnected organelles is called the endomembrane system and includes a distinct variety of organelles that have distinct functions. The nuclear envelope, right here, the Golgi apparatus in blue, the endoplasmic reticulum, then the Golgi apparatus in red, and of course the plasma membrane, as we see here, okay, and the vesicles. So all of these little tiny membrane enclosed structures here and here, and you can see some even fusing in the plasma membrane, are all vesicles. So this is an overview of the endomembrane system. Having seen the basic outline of the endomembrane system, we can therefore say that the endomembrane system in eukaryotic cells can be viewed as one large, dynamic, integrated, interconnected, and coordinated functional membrane unit operating inside the cell 24/7, all year round. So no breaks unless it hibernates or it dies. So there's no man-made factory that comes even close to mimicking the capacity of the cell to manufacture, to recycle, and move goods around. So the question then is, how are they specialized in their functions? So first, the endomembrane system makes two compartments within a cell. First, the area within these organelles, and then the area outside of these organelles. So this is the special dimension that we need to pay attention to. Second, this separation allows distinct types of biomolecules to be sequestered, to be organized, and then processed in each organelle, and therefore specific functions to take place first within the space that was defined or that is defined by the membrane, as well as within the membrane of the organelle itself. Okay. So therefore, this sort of functionally defined separation with interconnected and communicating organelles are akin to distinct but coordinated departments and rooms in a large and complex chemical factory producing all sorts of chemical goodies in precise sequence. Now, you may ask the question, where are these chemical goodies from the endomembrane system delivered? There are many destinations within the cell, but a major destination point is, of course, the plasma membrane. So, in the process of delivering goods in the plasma membrane, in the process called exocytosis, a vesicle that has budded off from the endomembrane system, like so, over here. So, it is budded out from the Golgi apparatus in this figure, can fuse with the plasma membrane and so deliver its contents, spilling the contents out into the extracellular space. So this process that takes place if cells would, for example, like to secrete a manufactured or processed product such as hormones, or even a replicated virus particle like COVID-19, for example. Now, the reverse can also happen. This process works in reverse in a process called endocytosis, whereby the material from outside the cell is brought into a vesicle, like so, and buds off inside the cell and gets fused to the and Golgi apparatus, and with even with other organelles, for example. So, for example, it can fuse with, let's say, a lysosome. This process takes place when cells acquire food particles, for example. So when cells come seeking for food, that they can use the mechanism of endocytosis, or if the cell gets hijacked by an infecting virus like COVID-19, then the virus can enter the cell through the process of endocytosis. We can have a look more closely into this process, in fact, in some future date. Now let's look at the individual compartments or organelles in the endomembrane system. One of the major objectives of this section is for you to be able to explain how nuclear envelope regulate the movement of molecules in and out of the nuclear compartment. So the nucleus, as shown here, and this is a figure, electron micrograph figure of the membrane. This the process that they use is what we call the freeze fracture, where you can actually um, slice open a frozen piece of the membrane and see what's in there. And so what you see is the um, uh, membrane of the nucleus. Of course, the nucleus stores the cell's DNA and is the site for transcription and RNA processing. It's also the site for DNA replication, although, as we will see later on, DNA replication happens when the nuclear envelope has disintegrated. But that will be for another chapter in this unit. But the important focus here is the nuclear envelope, which defines the boundary of the nucleus. So this is actually a figure of that. So the nuclear envelope consists of two membranes. So it's very interesting to take note of that. There's an inner membrane and outer membrane. Each of these membrane are made of the usual phospholipid bilayer. What is intriguing is that as you look at this figure, the nuclear membrane is perforated by protein openings called nuclear pores. Very important for you to remember that, which allow and control molecules to move into and out of the nucleus. So this is the key structure that allows the nuclear envelope to regulate what sort of molecules can come in and out of the nucleus. Okay, what gets imported and what gets exported. Small molecules and ions can passively diffuse through the pores, but large proteins and RNA requires active transport, the expenditure of energy to do so. These pores are essential for the nucleus to communicate biochemically with the rest of the cell. So now let's look at the endoplasmic reticulum. So the first thing that I would like to emphasize in the endoplasmic reticulum is that it is actually physically continuous with the outer membrane of the nuclear envelope. So if you look closely at the figure here, you can see an example of how the outer membrane of the nucleus is physically connected to the endoplasmic reticulum. So if you look at these schematics, the ER is a large organelle in most eukaryotic cells. It produces many of the lipids and proteins used inside and outside of the cell. And if you look at the configuration or the pattern of the ER, it is actually a network of interconnected tubules, like so, right? And discs, right? So there is this interconnection here. The interior space of the endoplasmic reticulum, and it's very important to remember this term, is called the lumen. Okay, and this is where a whole bunch of manufactured products are deposited. The convoluted arrangements of the membranes has an important biophysical significance because it allows a large amount of membrane surface area to fit within the limited volume of the cell, and therefore maximizes the surface area for the manufacture of all sorts of products. So for example, um, the endoplasmic reticulum has two divisions to it, the rough ER and the smooth ER. The rough ER is associated with the ribosomes as magnified here. So the ribosomes are bound to the rough ER. That's why the reason why it's called rough ER is if you look under high magnification electron microscope, you will see that the membrane is studded with granular objects, which are actually ribosomes. And these ribosomes are synthesizing or manufacturing proteins and directly delivering them into the lumen of the ER. And usually this happens if, let's say, the proteins are meant for secretion, if they are need to be secreted out, or if they are integral membrane proteins, for example, membrane proteins. Usually they get associated with ribosomes bound to ER. They are manufactured in ribosomes that are bound to rough endoplasmic reticulum. However, if proteins are simply needed in the cytoplasm, then you have, of course, ribosomes that are free in the cytoplasm and simply manufacture proteins and releases them there outside the organelle. So the rough ER is therefore responsible for manufacturing proteins that are meant for secretion or for integration into membranes. Um, and the smooth ER, on the other hand, is the primary site for lipid synthesis. So here is a blow-up figure of rough endoplasmic reticulum with the ribosomes manufacturing either secreted proteins or integral membrane proteins. And here is a blow-up figure of a smooth ER. You notice that the smooth ER lacks ribosomes, and the portions of the smooth ER bud off to produce vesicles that are free to move in the cytosol. So these are vesicles that are budding off. The smooth ER is, of course, the site of fatty acid and phospholipid synthesis. And therefore, you might expect that smooth ERs predominates in cells that specialize for the product of production of lipids. So, for example, cells that synthesize steroid hormones have a well-developed smooth ER that produces a large quantity of cholesterol, which is, of course, a derivative um, that is converted by an enzyme to steroid hormones. So the next step for a vesicle that is budding off from an endoplasmic reticulum would be the Golgi apparatus. So it is important to note that the Golgi apparatus is not physically attached to the ER, okay? But it is communicating or interconnected with the ER through the fusing, the budding of and fusing of vesicles. Okay, and this can actually happen in reverse. Now, the Golgi apparatus has three functions. First is to further modify proteins and lipids that were produced in the ER. And then, second is that they sort proteins and lipids as they move to their final destinations. I always consider the Golgi apparatus like the Amazon warehouse where you kind of determine where the destinations of molecules are, and then you put address codes in them. And in fact, what happens during the modification process is that the modifications of proteins and lipids serve as a molecular tags to designate where these proteins would ultimately be delivered for their final destination. And the third function of the Golgi apparatus is to synthesize the cell's carbohydrates. Of course, we know that carbohydrates are very important, a major biomolecule in living cells. So let's have a closer look at the structure of the Golgi apparatus. The Golgi apparatus consists of a series of flattened membrane sacs called cisternae. And they are surrounded by obviously by small vesicles that are either budding on, fusing in, or budding off. In this figure, we have two distinct pathways for two distinct types of proteins. One is for the movement of soluble proteins from the main source coming from the endoplasmic reticulum, and it gets modified through the series of cisternae in the Golgi apparatus, and from each cisternae you move the product, the soluble proteins, through a vesicle to the next cisterna, which is singular for cisternae. And as it goes from one um, cisterna to another, there is an increasing level of modification of the soluble proteins until it gets fully matured, and then by the process of exocytosis, then fuses with the plasma membrane, releasing the soluble protein. Whereas a transmembrane protein, an integral membrane protein, in other words, also coming from the endoplasmic reticulum, gets delivered by the budding off of a vesicle and fusing in the endoplasmic reticulum. I'm sorry, in the Golgi apparatus, and then goes through a process of series of modifications. And typically, transmembrane proteins gets glycosylated, or the additional carbohydrates are added to their structure before they get and fuses with the membrane. And since because they're integrated membrane proteins, they stay there as part of the um, uh, plasma membrane structure for to act as an integral membrane protein to perform a certain function, like, for example, pumps or transporters or other functions. Okay, so you can see, therefore, how important the Golgi apparatus is in terms of modifying proteins. And of course, not shown here are lipids before they get to their final destination. I must add as a final point to this slide that the modification processes that the proteins and lipids go through in the Golgi apparatus are all mediated by very specific types of enzymes as they progressively, as these products progressively go through each of the cisternae of the Golgi apparatus. So you may ask the question why proteins and lipids have to go through complex modification through the Golgi apparatus. And of course, the general answer to that is that these modifications provide additional specificity to the structure and therefore the function of the proteins and lipids that are being manufactured. So in the case of transmembrane proteins here, illustrated here, this process is called, of course, glycosylation. And this is a process whereby sugars are covalently linked to either lipids or to specific amino acids of proteins as they move through the Golgi apparatus. Now, these glycoproteins, as they are called, protect the proteins on the cell surface from enzyme digestion by blocking access to the peptide chain. So they therefore form a protective and flexible coating to the plasma membrane. Another aspect that I need to emphasize also is this, as I think I alluded to this earlier slide, that the process can also be reversible, where vesicles move from Golgi apparatus to the endoplasmic reticulum. So this happens because membranes tend to be recycled, membrane components tend to be recycled or to be retrieved between ER or Golgi membranes that sometimes gets accidentally moved forward and therefore has to be recycled back into their right places. So this whole process of movements of materials between Golgi apparatus and ER and vice versa, these are very reversible process because of recycling functions. Speaking of recycling, one of the major organelles that are super efficient in recycling materials inside the cell are the lysosomes. We can, in fact, describe lysosomes as the recycling departments of the cell. They are specialized vesicles that are derived from the Golgi apparatus, right here. They degrade the damaged or unneeded macromolecules, or even organelles that are getting older. So the cells have an efficient way of recycling all of these precious materials through the lysosome. And there are two important events that happen in this process. One is the emergence of a vesicle that carries large molecules from, let's say, an organelle or even from a Golgi apparatus, merging with the forming lysosome. And the second is the Golgi apparatus can also deliver enzymes that it processed to maturity. And these enzymes are enzymes that degrade the macromolecules. Okay, so they are called degradative enzymes or digestive enzymes. So the combination of these two materials, the macromolecules that needs to be degraded and the enzymes that digest them, um, allows the formation of a mature lysosome organelle. And the examples of macromolecules are proteins, of course, nucleic acids, lipids, and complex carbohydrates. And the lysosomes, they actually contain specialized proteins in their membranes called the proton pump. And again, you encounter this word pump. It means that this is a transmembrane protein that uses active mechanism to deliver protons into the lysosome and allows it to have a really high concentration of protons or a very low pH. In fact, it maintains the inside or the lumen of the lysosome into a pH of 5. And this facilitates the acid breakdown of large molecules. Um, and then the broken down molecules are then transported out by way of integral membrane proteins called transporters, right? So all of these important processes of recycling can happen in organelles called lysosomes. It is important to remember that the lysosome must maintain its integrity, because if there is any leakage of the membrane, then that will be very harmful to the rest of the cell, because outside the lysosome, the pH is normally um, around 7 or 7.5 in that region. So that it is important for, therefore, for the lysosome to maintain this pH 7 only inside it, and not to allow these protons to leak out. So this is the function of the lysosome. So this is the end of part 1 of section 5.4. So before we leave, I would like you guys to answer this brief question. Cells that line your intestines are known to possess a large number of membrane proteins that transport small molecules and ions across the plasma membrane. Now, which of the following cell structures would you expect to be required for this function of the cells? A, the endoplasmic reticulum. B, peroxisomes. C, lysosomes. Or D, the cell wall. So please take a moment to answer this question in your brief account. Thanks. I'll see you in part 2 of learning objective 5.4 or section 5.4. Thank you.